Hi guys, this is Kendall Terry, and this is part 2 of population evolution, or population genetics. And we stopped looking at Hardy-Weinberg. We stopped looking at the fact that Hardy-Weinberg has 5 principles that have to be met for a population to be in Hardy-Weinberg equilibrium, and the fact that that really doesn't occur much in nature because of that. So let's look now. So what takes place then in a population is called microevolution, that population changing over time within that population. And so we got to revise a definition then for us to really go from here. So evolution is a generation to generation change in a population's frequencies of alleles. Remember, Hardy-Weinberg deals with the frequencies of the dominant and the recessive allele and how that frequency changes over time. That's what we're looking at. So we've had to revise our definition there of evolution to fit what we're talking about. There's a couple causes of this. The first is genetic drift. And genetic drift is a change in a population's allele frequency due to chance. Now normally, if you have a large population, it takes chance out of it a little bit. There's still chance in the reproduction, but it causes that frequency usually to balance out. If you flipped a coin, let's say, for 100 times, if you got 80 heads and 20 tails, you would probably wonder what's up with this coin. Why is this coin flipping that way? Because it should be 50-50. However, if we reduce that down to just 10 times of flipping a coin, we would still expect to get a 50-50 split on heads and tails. But instead, if by chance we get a 70-30, then the results drifted in one direction because it was only a small population. It was only a small amount of flips, and that small amount allows that drift to take place, and we don't question that. We would say, you know what, I only flipped this coin 10 times. Well, in a population, the same thing happens. If we've got a small population, we either only start out with a few parents or we only get a few offspring, and the population drifts in frequency. That's called genetic drift, and that can change that population. Over time, we can start to see that allele ratio change. Some others that deal with sample size being large or small. The first is bottleneck effect. In the bottleneck effect, we reduce the overall genetic variability in a population from a gene pool due to some type of natural disaster. This could be, let's say, a fire goes through and destroys a forest. That's going to change who's alive. So whoever's alive now to reproduce will affect the next generation. So you could see a change in your frequency due to some kind of natural disaster where only a few members survive and they then reproduce the next generation. That's called a bottleneck effect. You could think of that if everybody's in a bottle and you pour out 10 individuals out of that bottle. It's a random choice maybe of even who's poured out. But those 10 individuals then reproduce the next generation. Your frequency is probably going to be different. Similar to bottleneck effect is called the founder effect. In the founder effect, you have a few individuals from a larger population colonize an isolated island or lake or some other new habitat. This happens sometimes in rivers. You get a river where it bends back and forth, maybe a flood even comes through, and when that flood comes through, it cuts a new pathway, and one of those bends, they're called oxbows, one of those bends gets cut off, and now it's a lake out here. Well, whoever was in that area, whenever the water started to go back down, that got isolated over here, they're the ones now that make up that new population that's isolated from the larger river population. So you end up with a founder effect where you could have the frequency change very quickly from one generation to the next because you reduce the number of offspring, or you reduce the number of parents, and thus you have a different ratio. Another cause of microevolution is natural selection. Remember, that was one of the 5 that Hardy-Weinberg had talked about. Results in alleles being passed along to the next generation in numbers disproportionate to the other relative frequencies in the present generation. In other words, somebody has something that makes them more adapted or more able to survive that generation, or just more likely to reproduce that generation. So that's natural selection. When somebody is better suited to the environment, they survive better, they reproduce more, possibly, whatever may be the case. So the gene frequency then changes. they'll be more heavily represented. Let's say if you have flowers, you have red flowers and white flowers. If the pollinators, we could say birds in this case, choose the red flower over the white flower more often, then the red flower passes its genes down to the next generation more often than the white flower. That's natural selection taking place. And the next would be gene flow. Gene flow is caused by gaining or losing alleles due to migration. If you're in an area and you have a lot of individuals coming in or coming out of your population that you're trying to monitor, they're carrying alleles with them. So if they come in from another population, they're carrying the alleles from that other population into this one. That changes this population's allele frequency now because every individual has to be represented. So if you have large migrations either in or out, that's going to change the gene flow, and that'll change the variability of a frequency in a population. The final one that we're going to look at is mutation. And in mutation, that's a change in an organism's DNA. So if you have mutation taking place where certain genes are being altered, the DNA is being altered, and that gives a different phenotypic expression, a different genotype, then now you've got a different, you've got something else in the population you have to consider. So if you've got mutation taking place, that's going to cause your population to change over time as well. The next thing now we're going to look at is, okay, now we've looked at what causes microevolution. Now we need to analyze microevolution. The first thing we look at is the fact that we have a diploidy effect. Most of the organisms that we're looking at for the course of this, course is that they're diploid organisms. Diploid organisms allow for less favorable alleles, the recessive allele many times, to remain in a population. So if it's recessive and causes a problem, it could be hidden in a heterozygote, where it's got the dominant allele, so it's fine, and it's masking or hiding that recessive trait that would be problematic. So that allows that recessive allele to hang out from generation to generation. And it's like, why is this not being weeded out? Well, maybe the homozygous recessive ones are being weeded out, but because of the heterozygote, because of the diploidy effect, that recessive allele will continue to be passed down from generation to generation, and you'll see that in the frequency. Another thing that we can look at in microevolution is a heterozygous advantage. We see this in the case of sickle cell anemia. In sickle cell anemia, if you're heterozygote, you have the ability to fight off malaria. If you're homozygous dominant, meaning you don't have the sickle cell allele, then you're susceptible to malaria. If you're homozygous recessive, you have sickle cell anemia. Both the homozygous dominant and the homozygous recessive will be weeded out to some degree. So the heterozygous ones are the healthiest ones in the population. So they have a heterozygous advantage. If they have a heterozygous advantage, then they're more likely to reproduce the next generation, and you end up with both of those kind of balancing the effect of the recessive allele out. Even though that does cause harm, the heterozygous advantage keeps that ratio high. The next thing to look at when analyzing microevolution is the Darwinian Fitness. This is a contribution an individual makes to the gene pool of the next generation relative to the contributions of others. How fit are you? Well, that depends on how successful you are. How successful are you at surviving? How successful are you at reproducing? Not just how successful you are, but how successful are the others around you. That gives us what we call a relative fitness, which is an assigned number based on genotypes of individuals in relation to success of the other individual genotypes. For instance, let's look at this example. The red flower plants, let's say, in our fictitious population we've been talking about, the red flower plants produce 10 seeds. So they survive and they produce 10 offspring. Their relative fitness for this population is a 1. The white flower plants, for whatever reason, only produce 8 offspring on average. So the red flowers are producing 10 offspring on average. The white flowers are producing 8 seeds, or 8 offspring. So their relative fitness is 0.8 because they're not as fit or successful as the white flower. So over time, you will see a population evolve because you have a relative fitness difference. One is better fit for their survival than the other. The last thing here is as an environment, or analyzing is as environments and selective pressures change, so do the populations. So we've got a couple graphs to describe the type of changes that can occur in populations. So if, let's say for instance, we're looking at fur color in mice, and we have light mice on this end, dark mice on this end, right here would be that heterozygous individual. So we see a population, on average, this would be the fictitious perfect population. It's got a nice bell-shaped curve. Frequency of individuals in a population, high in the middle, low on the ends. But let's say there's some kind of environmental or selective pressure, where in this case, directional, all of one side of the bell curve is being removed. And that could happen either way. I've got it on this side, which would be the light-colored mice are starting to be eaten more often for whatever reason. That is a directional selection, because if they're eating more, then they're not around to reproduce the next generation. And what you find is that your population shifts to where your bell curve comes this direction. The next is a diversifying shift. In a diversifying shift, the heterozygotes are the ones that are being selected against. So they're being weeded out, which gives you more individuals here and more individuals here as the population changes due to the heterozygotes being removed. That's called diversifying. The last one is called stabilizing. In a stabilizing population, this is actually what we see with sickle cell anemia. These are weeded out, these are weeded out, and you have a heterozygous advantage. So you have more here than on the end. So your bell curve becomes very skinny with high in the middle, low on the sides.